about today is the overall picture of biodiversity dynamics. And so what I wanted to do, I was initially going to just talk about islands and evolution on islands, which is my main research. But I just wanted to talk to you a little bit also about some of the more exciting things that we're doing on campus. And this is through the Berkeley Natural History Museums. And I'll talk to you about the overall picture of biodiversity dynamics, just understanding the history of biodiversity and how it might change as we go into the future. So with this kind of general idea in mind, the campus started what is now known as the Berkeley Initiative in Global Change Biology. And this was something really spearheaded by the Vice Chancellor for Research here, Graham Fleming. And the idea was to couple knowledge of past response to climate change with the analysis of contemporary evolutionary and ecological processes. So to put this information together so that we might be able to predict the vulnerability of species and the kinds of ecological and evolutionary processes that might be important for sustaining diversity, and whether we can use that information to kind of figure out where the best strategy as we go forward. So the idea here is that we've got this series of collections, of historical collections on the Berkeley campus, including, as Steve said, the Essing Museum of Entomology that I'm responsible for. There's also the Human Evolution Research Center with Tim White at the head, the Museum of Vertebrate Zoology, the UC Jets Herbaria, and the UC Museum of Paleontology and the UC Botanical Garden. All of these represent collections that go back over a long period of time, over 100 years. And so they have that history of collection and how change has happened over that time period. In addition to the collections, Berkeley has, well, the UC in general has a whole series of reserves across the state of California. And each of these are very rich biologically and serve as places where we can do research and where we have done research over a variable amount of time. So we've got the museums then that have this tremendous record of response to past climate change, and also they go back in deep time with the paleo record. And what we can do is then integrate the information across these different museums and bring together researchers in different departments to focus on those collections. This has been our idea. Coupled with that, then, we've got the field stations and that have been examined in terms of forestry, fire, grazing, and disease. And so they have a history of intensive research over the last 100 years, variably depending on the age of the field station. So what the idea then was to use these resources and couple these resources with the expertise that we have in computational biology, conservation biology, developmental biology, evolutionary biology, ecology, genomics, biomechanics, agriculture. Berkeley has this tremendous wealth of expertise in all of these different areas. So the idea then was to build this program that capitalized on all of these different aspects that the campus has. And so through funding from the campus, as well as initially from the Moore Foundation, we got a lot of people together to focus on these resources. And I just want to give you just a couple of snippets that have come out of this. These are just the main people involved, and I'm just going to highlight a couple of them. So I'm there with Charles Marshall, who's in the UC Museum of Paleontology, and we're really we're responsible for coordinating this. And just to give you an idea of a couple of the efforts that are going on. This is one that's led by Cindy Louie, who's in the UC Museum of Paleontology. And Cindy has been working with a bunch of different people, with Richard Dodd, Tony Barnowski, Kip Will, Brent Mishler, Roger Byrne, and Charles Marshall. And what she's been doing is doing a core into this, into Clear Lake, which is a bit north of here. And trying, by doing that core, she's getting all sorts of organisms that have lived in that area, as you core down over the last 150,000 years. So basically what she does is dig a core, and you get these quantitative records of change at this one site at Clear Lake over the 150,000 years. And then couple that with lots of little bits of information that we've accumulated across California, just to get a broader representation of change across the state, and integrate this data to try and understand how change has happened over the state over time. So what they've done at the moment is just get the core, and what they're doing now, this is just a picture of what them actually doing it. They just did it just a few months ago, and so this is basically what it looks like, and it's got these layers that include pollen, diatoms, insect remains, mosses, and all sorts of different things, isotopes, so you can get a picture of how change has happened. So that's just one effort that we're coupling the collections with this expertise to try and understand change over time. Another effort that I'll just highlight here, this is just a smattering of them, just to show you the kinds of things that we can do. So this is an effort that's led by Craig Moritz and Eileen Lacey, coupled with various people, Rasmus Nielsen, Mike Eisen, Maggie Kelly, Todd Dawson. What they're doing is focusing on one area and the collections that have been made in this one area. And it's a transect through Yosemite that focuses on these little squirrels, these chipmunks. And what they're doing is getting genomic and stable isotope information from chipmunks that were collected 100 years ago and chipmunks that were collected today. And they're comparing chipmunks, the alpine chipmunk that has decreased in abundance over the century, and one that has not changed at all and might even have expanded its range. And so this is a complicated and busy slide, but what I want to show you here is, if you see, this is up here is the alpine chipmunk here. Here you see the range has contracted in this one. This is the studiosus. It has not changed its distribution at all. So what they're doing with the collections of the alpine chipmunk, the collections of the studiosus that hasn't changed, they're trying to figure out signatures of adaptation that have allowed one species to adapt and according to the change in the environment, and the other species to weather it out, to contract its range. So they're looking at signatures here. They're doing this genomic analyses of the historical samples and the recent samples, and they can get really detailed information from historical samples. This is just showing you that the historical samples actually provide better genomic data than the recent samples. And they can pick out, just by comparing, this is historic and recent samples, and they're just looking for signatures of selection when you compare the two sets of samples. And so this just shows you the power of the historic and recent sampling to understand how things either adapt or don't adapt over the century. The last project I'll just highlight here, Neil Tsutsui, who works on bees and ants. He's been working here with Todd Dawson, Claire Kremen, Gordon Franke, and Santiago Ramirez. And what they're doing is they're getting the bees together with the pollen that they collect. And so what that allows you to do, then they're getting the current bees with the current pollen, comparing this over the century to ones that were collected 20, 30, up to 100 years ago with their 100-year-old pollen, and trying to see if there's a signature of change both in the bee and in the pollen. And so just very briefly, basically the answer is yes. This is on the left. You just see the signature of isotope change, that this is from the past to the present, and you get a very marked change in the signature of the isotopes collected from the pollen that these bees were getting over the course of the century. So this is basically comparing the historic bees and the current bees and giving us a picture of how the environment has changed over time. So with that information, we're going to be able to go on and build much better predictions of how things are going to change as we go into the future. The problem, though, with all of this is that it's a huge amount of information, some of it good, some of it bad. And we're trying to put all of this information together to try and get a detailed picture of change over the past. And this is where, again, through this Global Change Biology Initiative, we've been working with the Keck Foundation. And what we're doing here, this is another little area of the same overall effort to try and understand change over time. But this one is focused specifically on information. And so what we're doing here is trying to integrate information that you get from collections. So from all the museums, like these are the collections that we have here in the Berkeley Natural History Museums. And these are collections that we have from the field stations and various other little bits of data that we have from specific sites. What we want to do is get that data and put it together with data from layers that GIS people get, so layers of vegetation, climate, rainfall. And the idea is to integrate this information from the specimens and from the layers of vegetation or rainfall or whatever, and put that together and try and get a better representation of how change, genotypic, phenotypic, adaptational change is happening over time. So this is a very ambitious effort, but I'll just tell you roughly where we're at with it and how it's going. So the first thing is getting the specimen data from the museums. And you think, well, that's fairly straightforward. And certainly for some of the smaller collections, it is. For the Museum of Vertebrate Zoology, they've got all of the data in digital format, so you can access all of that data associated with the vertebrates. The Museum of Paleontology, they're working on it. The Jepson Herbaria, the UC and Jepson Herbaria, they're also working on it. But for the insects, it's still an issue. The problem is that you've got all of these specimens here, and each of these specimens, here you just see a tray of butterflies. Each of these specimens has information on where it was collected, when it was collected, the kind of environment that it was collected in. We want to get access to that information. And how do you do that? The information is actually underneath each of these specimens, so you have to peer under the specimens and find it. And there are millions of them, millions of these things. So at the moment, we've currently digitized just 150,000 of the 6.5 million specimens just in our museum. And that's less than 1% of the specimens. So what we've been trying to figure out is how do we speed things up? Yes?
When you say you digitize the specimen, what information do you include?
So that's an excellent question. So what we want to get is information. We want to put in information about exactly what the organism is and where it was collected, so that we can try and figure out how the distributions have changed over time, and all of the associated data, like the abundance of this particular invasive moth, for example. It only appeared in collections, say, in 1963, and since then it's just burgeoned. Can we, if this is the kind of information we should be able to see there, and coincidentally, we should be able to see increases in one with decreases in another. And at the same time, we're going to put in information on how the ecology, the phenology, all of these kinds of things that are associated with that one organism, and how they have changed over time to get a broader picture. Yes? That is the biggest, that's the nail on the head. So that is actually the biggest problem of all. Some have been collected systematically, and we're targeting those initially, just to try and see whether you get actual changes over time. Most, as you could probably see, have not been collected systematically. even those that are, different people are going out, so there are different collecting methods. So we've got this group at the moment that are actively working on what you can do with incomplete data. I mean, at some level, you pull over both space and time. And at how much pulling do you have to do to get any signature out at all? So this is the kind of, there's a statistical group that are working on the first part of the project who are trying to figure out just what data whether there is anything we can get out of this. I mean, at some level, especially as you come towards the present, where we are systematically getting this data, we'll certainly get a lot out of it. And for certain questions, like the Grinnell Resurvey project, we'll also get some specific answers out. The more broad-scale use of the collections and figuring out change over time, we'll get something out of it. Whether we'll get those distributional changes at what level, we really don't know. But the idea now is to get this in digital format, and it's that so just so that we know what's there, when they were collected, and what we can do with it. So this is actually where we come to a bit of astronomy. So what we've been doing, at least in the biggest problem with this is the entomology collections. And so we've been teaming up with the Adler Planetarium in Chicago, and they were responsible for developing what's being called Galaxy Zoo. I don't know if you've heard of this effort, which is a big citizen science effort. And the idea is that you've got a lot of images of different galaxies out there, and the more eyes that can look at a given image at a different time, the better. And you get errors associated with lots of people looking at a given image and categorizing it according to the type of. I feel very bad about talking about this stuff to people that know a lot more about astronomy than I do, so I won't say anything more. But it's a huge effort and tremendously successful. And through the people at the Adler have now developed lots of different efforts. Galaxy Zoo is just one. Oops, sorry. But there's the old weather, where they've tracked weather records over the ocean over the last 100 or 200 years, and they get a very accurate assessment of how temperature has changed over the ocean. But again, just using people out there to look at these records and put them into databases. So what we have been talking to the Zooniverse people about is doing the same thing for specimen data associated with the collections. So Arfon Smith, who's at the Adler in Chicago, and Andrew Hill, who's with this company Visuality, they've been working with us to try and see how we could get this kind of information that you see up there that's associated with this beetle. How do you get this into a database? And how do you, when you can't just kind of put an image up there and say, oh, come people, just get that into a database for us. You need to engage the public. You need to get them involved in what they're doing. That part of this is an educational initiative so that the general public can see the value of getting this information into a database so that it can be searched and integrated with other pieces of information. So that's just one of the big issues with getting this information digitized. So once we have this data in a database, in a searchable format, what we want to do then is integrate the information across these different collections, across the Museum of Vertebrate Zoology, the entomology, the herbaria, the paleo collections, and put this together with data that's been published and the genomic data. If we can get all of this data together, then we can start to get a better picture of change over time. So this is, as you can imagine, it's a big programming effort, and these three people at the bottom, Ginger Ogle, Joyce Gross, Michelle Koo, they're all major programmers who are really into this. But I'm just going to say that they're doing a tremendous job right now, but at the same time as they're doing the work on the big work on getting the layers together, the layers of vegetation, precipitation, and things like that. Maggie Kelly and Kevin Coy here with the GIFT facility on campus, the Geospatial Innovation Facility, they've been putting this together. They developed a program called CalAdapt that allows you to see how layers of precipitation, temperature, etc. have changed over time. And they've got layers that reflect hydrology, soils, etc., etc. And so there's all of these layers of data, and then the real challenge, of course, is to integrate these, the point data and the spatial layers, so that you can get some idea of how organisms have responded to precipitation change, temperature change, etc. So basically the idea is putting these point events associated with given collection events, etc., and plonking them down onto the spatial layers and getting a better picture of how change has happened over time. And this is a very boring slide, but it's just basically, Kevin just came up with this just 2 days ago. And it's very exciting because it shows that actual progress has been made in terms of both integrating the different kinds of data and integrating with other efforts that are going on elsewhere, and just figuring out that's a clear pathway as to how we get information out and how we get it to where it can be kind of hooked into other programs and analytical tools. So that was just a whoosh through this effort that we've been doing, the Global Change Biology Initiative. What I want to do now is just tell you a bit about what my specific research is and how it relates to the whole issue of change over time. What I'll be doing now, just to start off with, is going back a little bit in time. So we're now talking in more deep time as to how change has happened as you go back millions of years, but within an identifiable chronological time frame, as you'll see in a second. So what I'm doing is looking in the middle of the Pacific Ocean, and here you see the islands of Hawaii that are stuck way in the middle of the ocean. You often don't realize just how far and out they are, but they're extremely isolated little islands out there, and because of that, very little has been able to get to these islands. But one thing that has got there are spiders. And so you think, how did spiders ever get to these islands? And here's just shows you how spiders disperse. And what they do is they go up to something high, let out silk. You see it's putting its little abdomen in the air. You see silk is coming out, and the wind pulls and pulls, and then poof, the spider takes off. You see that here's the wind pulling again, and off it goes. And so this is the primary mechanism that spiders use for dispersing. It's called ballooning. And so this is presumably how they get to these very remote islands. So here you have Hawaii. So why is Hawaii so wonderfully spectacular for looking at evolutionary history? And the reason is, first of all, that it's supremely isolated. It's the most isolated archipelago in the world. But also, and because it's so isolated, very little could get there, at least initially, before humans got there. So it's not only very isolated, meaning that very little got there, it's also arranged in this chronology from these different ages of the islands that you see here. So it goes from the youngest island of Hawaii up to the oldest island of Kauai. Basically what happens is that the lava oozes up under the ocean. And so it oozes up, forms these volcanoes. The volcanoes move away from this hotspot that's being formed. And as the islands form, they become high, move away from the hotspot, they become eroded and go down and eventually disappear. So within this kind of framework, you can actually see the different stages of evolution, with the most mature stages on the oldest island, the youngest stages on the youngest island. So this kind of framework has been the basis for some of variations. And this is just one of them. These are the Hawaiian honey creepers, which are very similar to the Galapagos finches. But if you know about the Galapagos finches that Darwin talked about, he talked about how their beak was modified to feed on different kinds of food. In Hawaii, you have the same group that colonized the islands. They diversified in the same way, but the beak shape is just much more spectacular. And the reason is simply that they're bigger islands and they're much more isolated. So very little else got there. So basically these birds took over most of the aerial niche for birds. There were others, but most of them, I won't talk about them right now. The other groups that have done a similar thing, this is work that Bruce Baldwin, who's here in integrative biology, he's been working on this silver sword radiation, which is put in several different genera, but they're basically the same lineage, but they look so very different. You've got the big silver swords, the little shrubby things, the little floret things, and big trees, and they're all very closely related. So just a big radiation of things that got there and just went all over the place. One of the best known of the adaptive radiations, as these are called, of organisms in Hawaii is that of Hawaiian flies. And these here, this is a tree, and I'll talk about these in a second, but it just shows relationships amongst the flies. And these are, on the left, they're host associations, and you can see their major host associations have changed kind of low down in their relationship tree, and then they still kept speciating. And the reason that they seem to keep speciating is that they've got this very elaborate courtship behavior. And if we just see here, just so you can see, this is a male on the closest to me, and he's dumping pheromone on the head of the female there, and she's deciding whether or not he's good enough to mate with, just based on what he smells and tastes like. And so it's a very elaborate courtship behavior, and it seems like speciation is really on the basis of this elaborate courtship behavior. So the speciation there happens really fast. What I've been doing is working on spiders, and in spiders, what's nice is that they're very stuck in an ecological environment. They often build webs, and the webs tell you basically where and how they want to live. And then they come out, you can go and collect them and watch them. They don't see that well, except for jumping spiders, and I'm not going to talk about jumping spiders. But these ones they're very easy to work with and understand how they live and why they live where they live. So let's just think about just a little bit of history as to how I ended up working in Hawaii on these things, just to give you an idea what the environment's like. When you think of Hawaii, Steve, it might be different because you go to the top of the mountain and you get a better feeling for what's in between. But when you think of Hawaii, you often think of the beaches and the palm trees, and as Mark Twain said, the loveliest fleet of islands that lies anchored in any ocean. The thing is, though, when you try and work in the forests of Hawaii, it's kind of a different story. And yes, they're beautiful, but gosh, they're hard to get up to. The terrain is really steep, they're very, very wet, and access is also quite difficult. And so when I started working there in the mid-80s, it was actually very hard to get anyone to go with me, and all of these agencies required you had to go with someone. And so I found my mom. I brought my mom from Scotland, and so she was great. She came all through the Big Island with me and over on Maui. But then, anyway, that's a long way between Scotland and Hawaii. So I found someone else who would come with me, and this is in the early 90s, and this is George Roderick, who's in the department here, very quickly became my husband, which made life a lot easier. And then we had offspring who also, well, he wasn't very useful at that age. But anyway, he got steadily more useful as time went on. He could actually carry the net and carry something. He looks like he's not enjoying himself, but they got a lot better. So this is kind of the history of how of work there. But what was so exciting? What made me want to stay there? And this is what I want to tell you about. So we've got the islands then, and as I said, you've got this arrangement from older to younger islands. What organisms have tended to do, the ones that have been there for the longest time, they've tended to hop down from the older to the younger islands. So let's look then at patterns of diversification within lineages of spiders. So I'm going to tell you about the spiders, and I'm going to tell you how they're related across the islands. So a lot of what you're going to see is trees of relationships. And these are based on DNA sequences. And this is a very simplistic thing. I'm sure that many of you are familiar with how you do this. But basically, you look at sequences of DNA and then see how they've been modified. And you group things according to how the DNA has been modified. So this sequence is the ancestral one. And then one of this C here got changed to a G in 3 and 4. And so they were grouped together because both of them share that change. And in this one, this A got changed to a C, and so these two are grouped together because they share that change. And this is when I show you the trees that you'll see here. This is basically how they've been generated based on molecular data. So let's just look at some of the spiders then. This is work that I did with Gustavo Hormiga, who's at George Washington University. And Gustavo, he described this spider, and he liked Orson Welles' plays. And so he called it Orson Welles, and then all the species are named after the plays. But anyway, it's a really amazing spider. It builds really big webs. But it's undergone what is called really a non-adaptive radiation. It's basically hopped down the island chain without changing. It does basically the same thing. It builds a big sheet web and sits under trees, comes out at night, but hasn't changed adaptively. But there are a whole bunch of other ones that have changed adaptively. They've hopped down the island chain and diversified within the islands. And so this is what I want to talk about from now on. So here's just one. I'll show you a bunch of different lineages of spiders that have done this. These are stick spiders in the genus Ariamnes, and they have this incredible diversity that you see here. These are just the Hawaiian species, and just a kind of a representation of them. Some are really shiny, like you see in this one here, up in this one here. Some are very dull and white. This is what you get in the dry forest of East Maui. And some are dark brown. You get these down in the leaf litter. So the color tends to reflect the kind of habitat they live in. So what we did then was we generated a phylogeny and based on the DNA sequences and looked to see how that matched the geological history of the islands. And basically what it shows is they hop down the island chain. We can time this based on how much the DNA has changed. And you get this variation scattered across the tree. So what we did was, so they've hopped down the island chain, but you get that there's no real pattern in how this great diversity in color has changed. So what we did was we looked in the context of the color and the kind of ecology that they have in the context of this tree. And what's really interesting is this, what I've boxed out in the yellow here, I'll just show you just to see again. In the yellow, look at where the boxes are. These are these gold ones that live under leaves. And so they pop out all over the tree. And then there are brown ones, these are the brown ones, that live down in the vegetation. And again, they pop out all over the tree. So what this is showing us is that there's a clear pattern of evolutionary convergence, repeated evolution of the same ecological forms. So that's just one group of spiders. But then we started to look at this one big lineage. And these are the long-jawed orb-weaving spiders. And I'll just show you here, just to give you an idea of what we do here. So we collect these things. This is just a spider that, for whatever reason, decided to stay out in the day. And I'll see if I can get this to play or not. Maybe I won't get it to play. Yeah, you never quite know whether these things are going to work. It doesn't really matter. But it just to show you how they're collected, it doesn't matter. So they you collect these things and almost entirely during the night. And this is when they're active, they don't see you, and you can really understand their ecology. So what we did then was we've collected now just bunches of this one genus, and they're all very closely related, and they're tremendously diverse, all in this long-jawed orb-weaving spider genus Tetragnatha. So ancestrally they build these orb webs like you see there, but within those orb-weaving ones, there's just this gamut of diversity with tremendous specialization. This is just from under ferns on East Maui. This is just from the summit of Mount Kaala on Oahu. This is just the wet forest of East Maui. This is the lava flows of the Big Island. This one here just feeds on amphipods on the summit of the Kolas on Oahu. So they're incredibly specialized and tremendous diversity. You also get these ones here that you see with the photographs that David Lichwager and Susan Middleton took. These are the ones that you see just sitting out there. They don't build webs, and they represent another radiation that's just become a cursorial predator. So based on molecular data, what we found is they've diversified both within ones that just run around and don't build webs, this group here, and ones that do build webs and are still very diverse. And you get rapid diversification of both of these forms. So how do they diversify? And so I'll just show you just a couple of examples here. These are the web spinners, and what I had a postdoc, Todd Blackledge, who looked at the forms of the webs and measured all of the forms of the webs. And what he did then was he did principal component analysis on the web form, and he mapped that out in space. So basically, this is just representing ecological space. You can imagine it like that. So on the top left, this is one place on Oahu, and so this is one species that sits kind of here in ecological space based on the measurements of the web. This is another species that sits in this space, and this is another species that sits in this space based on the measurements of the web. Likewise, this is Maui, and you see they're well separated in ecological space there, and likewise in Hawaii, they're well separated in ecological space. So you see that you get similar web types when you bunch them all together. And so what Todd was interested in was seeing whether the similar web types were all related to each other. And here again, he found that was absolutely not the case. And so you get similar web types here, and then similar web type there, and again here and here. And so the similar web types, again, have evolved repeatedly. So convergence within web types. The final story I'll just tell you in this context, the spiny-legged ones, the ones that have abandoned web building. So here's a green spiny one, a big brown spiny one, a maroon spiny one, and a little brown spiny one. And you get green ones, and maroon ones, little brown ones, big brown ones in every habitat on every island, pretty much. There's some gaps. This is basically a picture of where they all are across the islands. There are gaps, and it's a longer story, but the bottom line is within any one habitat, you tend to get one of each representative of the big brown, little brown, maroon, and green. So we then looked at how all of these were related across the islands. What you find is a nice pattern based on all of this molecular data. You get a nice pattern of progressing down the island chain, just like we expect, from the older to the younger islands. But then, when we look at how they're related, what you find is that the green ones, see here I've just colored them, these are the green ones, all these green ones here. This is the maroon one, the big brown one here, and the little brown ones. And you see that they pop up all over the place. So here again, rampant convergence of similar forms. So the bottom line, what we've been showing here is that you get, within these lineages, you get rampant convergence to a similar array of ecological phenotypes on different islands. So basically, the ecological forms, like represented by this yellow, they pop up over and over again. And it's just the same set of forms that pop up over and over again. So what we're trying to do now is understand exactly how that happens. But rather than get into that, what I want to do is just tell you a little bit about what happens within species. And here I want to just give you a little bit of a story about the Hawaiian happy face spider, because this is a very cute little animal. And what's interesting about it is that it's got all of these colors. So this is one that's red at the back. But in any population, you tend to get most of them being plain yellow like that with black marks, but most plain yellow. But then you get all of this different array of red coloration superimposed on that yellow. The red front and back, red lines, red ring, red blob, and then even this pearly white one. And so you get all of these different morphs. And so the question initially was, why do you get such diversity? And what we did was initially we looked at the frequencies across all the populations that are found on all of these islands from Oahu down to Hawaii. And you find the frequencies stay remarkably constant in each of these populations. So why are they so constant? And what we said was either there's a lot of movement between the populations, or there's selection maintaining the color polymorphism within each population. So what we did, again, we used molecular methods and showed that there's actually no movement at all based on, they're just sticking to one place, very little movement. And so there's got to be selection maintaining the polymorphism within each population. And the most likely thing there is birds. Birds develop a search image that they're going to develop preferentially a search image for things that are most common in the population. So for the spiders, the most abundant will be the most cryptic morph, which you certainly find. The most abundant, most frequent morph is this plain yellow one that's hardest to see. And then the rest of the colors are all of this whole gamut of different colors. So you get this remarkable stability, presumably maintained by bird predation. We don't know that, but that's the best guess we have. But so why do you get this similar color polymorphism? Exactly what are the mechanics of it? And this is something that we're exploring now. We've done all of these crosses. This is work I did with Jeff Oxford. And what we did was we crossed spiders within Maui. We crossed yellow and all of these different colors. Yellow is recessive to everything else. So when you cross them with yellow with red blob, for example, the red blob will tend to be heterozygous for yellow, and so they come out an offspring half red front and half yellow. It's a very straightforward, clean, nice pattern of heredity in these things. So we did that in Maui. What we did then, Jeff was having so much fun doing this, he came over to the Big Island and did the same thing on the Big Island. What he found was, again, they segregated out in the offspring. If you cross a yellow at the top, you see the yellow here with a red front, half come out red front, half yellow. But the interesting thing is that the yellows on the big island are always female and the red fronts are always male. And then what's more interesting here, you cross a male yellow from Maui. Remember, you don't get male yellows on the big island, but if you cross a male yellow from Maui with a female yellow from the big island, the offspring will come out with this red front. So basically, what we think has happened, just looking back and saying, how could this have happened in the first place? So what we think happened was this. Here you've got Maui. They colonized the big island, and we've got information from that based on the molecular data. So they jump over, get to the big island, and when they did that, they would have done so with an incomplete representation of all the colors. So just one or two would have been involved in that colonization event. And then they arrive on the big island, build up a population. There would have been selection to recover the diversity of the ancestral population. So selection to recover all of this diversity. So the idea then is that there was convergence then to the same array of polymorphs within a species, but by a different genetic mechanism. So what we're doing now, and I'm not going to go into this at all, is what we're doing next generation sequencing, Illumina-based sequencing, and we're basically sequencing the whole genome of these spiders and trying to figure out exactly what changes have happened as they've colonized from one island to the next, and what the selective pressures might have been. So that's very much a work in progress, and we'll see how that pans out over the coming year. But before I finish, I just wanted to say, because you can't really talk about these islands without saying what has happened in more recent history. So I just want to talk just very briefly here about what exactly has happened and what might happen as we go into the future. So this is work by Pat Kirch, who's in the anthropology department here. And Pat has done some really fascinating work mapping the progress of Polynesians across the islands of the Pacific. So Polynesians started somewhere around Samoa and then colonized out into the outer regions of the Pacific very quickly and reached all of these very far-flung islands of New Zealand, Easter Islands, up to Hawaii, but all the way through, colonized all of these islands very rapidly. So what happened then was you get very rapid increase in the population size of the human population, and with that you've got a lot of warfare going on. So associated with that then, you can imagine the impacts. So in particular, notice this is just a painting, but it represents the kinds of things that they're wearing. And if anyone's been to the Bishop Museum, one of the prize capes of the kings was actually entirely yellow. And you say, well, that would have been a lot of birds, because it's all made of bird feathers. And certainly for the red birds, you can imagine the impact there. But the one that's all yellow is actually from a black bird that has just a tuft of yellow. So you can imagine the enormous impact it had on those birds, which are, of course, now extinct. And so, associated with the arrival of humans then, a dramatic loss of avian biodiversity. This is the work of Helen James. They're discovering these fossil remains in various caves on the islands. This is work from the Marquesas that David Stedman did, just documenting the species of birds that he's found in fossil samples, with the ones here in red all being extinct. So a tremendous loss of biodiversity with initial Polynesian arrival. Then, of course, Captain Cook came along with the best of intentions, and he mapped out the Pacific. What happened when he came was that he brought disease, entirely unintentional, of course, but with that disease, the population in each of these islands plummeted. Here you just see the blue is at the time of Cook's arrival to the islands, the population size in Hawaii, in the Marquesas, and Tahiti. Subsequent to Cook, the human population plummeted, And then since then, it's recovered. Of course, in Hawaii, it's much higher now. In the Marquesas, it still hasn't recovered. And Tahiti, it's exceeded what it was. But the bottom line is that there used to be a very large population, and now there's even a larger population. So the thing is, you've got organisms on these islands that have tiny populations, and so they're subject to the impacts of this arrival of humans with associated organisms. So the rate of extinction of, this is just documenting birds, the rate of extinction on islands is extremely high, and the islands of the Pacific are the highest. So if we just look here, this is just an example just to show you what's happened. This is on Oahu, this is just looking at Honolulu. But basically the outside world has poured into these islands. In Hawaii, you get more than 2,500 arthropods, 13 reptiles, 38 land birds, 19 mammals, 900 plants are now established, and this is just, it increases every day. There's land clearance, conversion. You get some amazing things going on. This is when things first arrive, they tend to reach very large populations. This is a movie. I don't know if you can get it to show. Yes. It looks like it's raining. This is getting off the ferry in Tahiti, and they're trying to see, well, where is the rain? And this woman is thinking, there's not a cloud in the sky. Where's the rain? And it's actually from these sharpshooters that invaded the islands and then reached huge densities that these invasive things tend to do. And it's come down a bit now, but they have a huge impact. So basically what has happened, here you've got Hawaii in the middle of the ocean, and what you've done is basically shrunk the Pacific Ocean, and it's now just an island that's very close to the mainland. So we've changed the dynamic going on in these islands. The isolation has been lost. So where does that put us as we move into the future? And this is what we just don't know. And this is something I'll just say here. I'm not, this always makes me cry. It reminds me, if anyone's read Dolores, when it says at the end, you think of the utter environmental degradation, and at the very end, when I read this to the kids, I just would always dissolve in tears. But it says, catch calls the once there, you let something fall. It's a truffula seed, it's the last one of all. You're in charge of the last of the truffula seeds, and truffula trees or whatever it needs. Plant a new truffula, treat it with care, give it clean water, feed it fresh air, grow a forest, protect it from axes that have, and the Lorax and all of his friends may come back. So the thing is, you think that is far too idealistic, and how is it going to happen? And so I'm not going to leave you with that, just in case you think you're going to leave entirely depressed. There is, just to show you that you actually can do something, this is an area that I'm with, there are kids a long time ago now, but this is in Oahu that's just been completely devastated. It's dry forest and it's been heavily grazed. And Art Medeiros, who was with the national park on the island of Maui, what he did was he basically got the community to go and just pull out the invasive plants. So he just got people to pull out the invasive things, and then they actually went in and planted the native stuff. And you think, that's a lot of work. But what's interesting, if you look on Google Earth, and you do this, go back home and you look on Google Earth, and you'll see, if you look at the south slope of East Maui, you'll see this dark patch, which is where this restoration effort is going on. So the forest has come back, and so here you see the forest just from a lower level, and you see, it's doing incredibly well. This is the inside of it. So whether the whole ecosystem's going to come back, we can hope, but it does show you that you can do something. And so I think the important thing that we can do as conservation biologists and people that are interested in preservation of the environment as we go on into the future is basically understanding what is there and getting the message out as to why it's interesting. And also recognizing that it's not hopeless and that you can actually do something. So with that, thank you so much, and I'll be happy to answer questions.
thanks so much. And I would say as well, Rosemary mentioned a couple of colleagues, Tony Varnosky and Neil Satsui, who we've also had lecture in this series. If you check our website, there's videos of their talks. And thanks to Chris and James at the back here. We're going to get Rosemary's talk up here in a few days. But yes, any questions?
Yeah.
So when I was seeing, you were talking about the species hopping down the islands and these recurring patterns, somehow I thought of, I'd heard of saber-toothed tigers. Somebody did a study where the saber-tooth would appear and then disappear and then reappear. And I got this idea that somehow the genes for saber-tooth were there, but they just weren't expressed in some times. And I'm wondering if there might be a similar thing going on here, where you've got the genes for coloration or whatever, they aren't expressed, and then they become expressed in the next topic.
That's exactly what people are starting to think now. And Mary Jane West Eberhard, who's a biologist down in Costa Rica, she's done a book on phenotypic plasticity and how that might play a role in facilitating adaptive radiation. So it's kind of this, the idea is that there's intrinsic variability in these organisms that can respond to selection. And so you get speciation within, trajectory A, B, C, and D. But it starts from an intrinsic flexibility to do that. And the reason you get, the green ecomorph or the maroon one, I mean, they look really identical on the different islands, yet they're not each other's closest relatives. And so it's got to be a mechanism like you suggest. With people working on sticklebacks have found a similar kind of thing, where you get an ocean-dwelling form coming into lakes. Repeatedly, they give rise to limnetic and benthic forms. And they're the same, the limnetic forms are very similar across multiple lakes, and the benthic forms, they do this over and over again as they've colonized freshwater lakes. And so it's again, the thought is that they've got this innate plasticity that allows them to repeatedly evolve the same form. So yes, that's exactly right. And we're trying to get at the molecular underpinnings of that right now. Yeah. Right. So that's, it's at one level it's straightforward, at another level it's not. So we are starting to get at this kind of information using genomic tools. You can get at how, in particular for chipmunks and things, you can get at what's changed. It's easier in chipmunks because there's a genome available for many of these different organisms. Work that Hopi Hoekstra has done on, she's at Harvard and works on these beach mice and forest mice. They've repeatedly evolved this blanched kind of white color through the same mechanism, and she's actually identified the gene that is involved in that switch that happens very, very easily. And so you can get at that whole mechanism. There's also someone here that, Bree Rosenblum, who's just arrived here, she works on chytrid fungus in frogs, but she's also worked on this blanching, this bleaching phenomenon in lizards in the white sands of New Mexico. And it's the same kind of thing, and they've identified it's pretty much the same gene that's involved across a lot of vertebrates. They have the same thing that switches on and off and allows them to change color. So the difficulty here, with Hawaii, you can actually see, what's nice about it is you've got this framework of islands that goes back into deep history, really much older, 5 million years. And so you can get an idea of how the whole thing might play out in the context of adaptive radiation. The trickier thing is just that the genomic resources available for these organisms have not built up to the level that they have in birds, most vertebrates. So it's still, it's getting there, and it's getting there rapidly, but it's slower than you might think. you're looking at a series of lizards or mice or something in Hawaii. If this were mice, we'd be there. We'd know exactly what genes are changing. But yes, that's an excellent question. Yes.
Is that a part of your research, and it apparently didn't happen on the Hawaiian chain of islands, but I'd really like to understand the island effect where there are species that are enlarged or
Where they get bigger or smaller. Yes, that's that's a tough one to get at, and so it seems like you know how this works in mammals in particular. They seem to get smaller. In invertebrates, they seem to get bigger, and then you saw one example of that in the Orson Welles. They're the biggest linniphiids in the world. Because linniphiids aren't the sheep web spiders. You see them all over here. They're just little things that sit in the bushes. These are substantial spiders, and they build webs that kind of go from the floor to the ceiling. So they've done exactly what you suggest there. I don't know why, and I don't. It's a tough one to get at exactly what the mechanism is. There are lots of different ideas as to why they might have done this, but nailing down those ideas is it's tricky, and especially working within an island chain, you'd have to work across the islands, and actually, Gustavo Hormiga is looking at this to some extent, at least looking at the evolution of the phenomenon, because he knows all about the ones in Hawaii, of course, but he's just been on a trip to the Juan Fernandez just off Chile, and you get the same, the gigantism in these linniphiids happens also in the Juan Fernandez. And so he's looking at the evolutionary history of that. Whether, it'd be again interesting to see the genomic basis of it, but exactly why they do it, and how general, it does seem surprisingly general, that the mammals always seem to get smaller. The idea is really that the mammals getting smaller, just the smaller space and smaller amount of resources. The larger size in the invertebrates, the idea is that they don't have as many predators. That's the way the arguments have gone, but they're entirely untested, and they're tricky to test. Yes.
How many spiders balloon to really cold climates and actually adapted enough to live?
So balloon to really cold climates, and adapted there to these temperate NASA, these ones that have got to Hawaii and diversified there, they're right. They're not in Antarctica, but they sure live in some just the coldest places and the hottest places that spiders live anyway. So yes, spiders can certainly adapt to incredibly cold places. They just what they do is in very high latitudes they tend to have a biennial cycle. So it takes them 2 years to mature, and they spend the cold periods just inactive. But they do surprisingly well. As long as there's just some kind of short period where they can get food, they do fine. I mean, if you put for most spiders, at least for most temperate spiders, you can put them in the freezer, the egg sac in the freezer, and they'll do just fine. So that they're pretty resilient. Yeah.
Do you think for conservation purposes, it's better to try to save these more ancestral species for the older populations, or do you think that doesn't really matter? Do you understand what I'm saying?
You're right. I know.
Is it best to try to protect the oldest islands, or?
It's a really hard question, because what we've done is superimpose basically an ecological process with all of these things coming in on top of this deep kind of evolutionary process that has given rise to this, repeated evolution of similar groups. So, to be honest, I think that the main thing that we need to do, besides just understanding what is there, is figure out exactly what happens when you juxtapose these two processes. If you go to some of these places, you think, how is that possibly going to work? There's an area on Maui that the Nature Conservancy protects, the Waikimoi Preserve, that they've done a really good job of keeping the weeds out of the reserve. You go outside the fence, and it's like an army of ginger that is just pushing at that, literally just pushing at that fence. And you think, how can this be sustainable? So I just think that if we focus on the details of the evolutionary process, I just think that we need to figure out more really what's going on in this ecological evolutionary interaction and try and see what can be protected there. But as I say, the understanding what's there is a big part of this, so that people can see what is worthy of protection. And in that regard, actually, Susan Middleton and David Lichwager have done an amazing job at just taking excellent photos just to show what's there. And when I got to Hawaii, most of these spiders were undescribed. Almost all of them were undescribed. And so you just you spend most of the time just describing and documenting what's there, so that at least there's more attention paid to these extraordinary the biology of these organisms. But I think the big thing is understanding exactly what's going to happen as you put the two things together. Yeah.
Roughly, how long is a generation, and how many generations before two species can separate?
Well, that's actually something that we're working on right now. We just what we're trying to do now is look at the youngest island of Hawaii, which goes from age 0 to almost 1 million years old, with a really nice juxtaposition of geological ages across the landscape. And what we're trying to do is see how the communities have come together and how they've evolved. So put together both ecological and evolutionary processes to see how that changes over the age structure of that young island. So to come back to the generation time, the spiders tend to live for a year, and so the generation time, even when you bring them in the lab, it's hard to population, just a kind of mosaic of repeated colonization, juxtaposition of different populations. But, how does that dynamic then play out into evolution? So we know we get these different pockets, but we know that that the lava flows over time will get forests and they'll come back together. The time of separation is not long enough for them to become isolated and genetically distinct. So does that fragmentation play any role? It surely fosters diversity, but does it facilitate isolation? Unlikely. And how does that all play out in the context of species splitting? And so I think the bottom line is I think it's not straightforward, but we're going to. This is something that we're trying to get a handle on. So we're working with this guy Rasmus Nielsen, who I showed was involved in the chipmunk project. But Rasmus is his focus is population genetics and uncovering signals from genetic data to try and figure out the history of populations and see exactly how they diverge.
So roughly the ecology makes it incredibly hard to understand evolution.
Right. So it's just that the geology is very complex, and what we're trying to, it's in the big picture. If you just say, well, this volcano is this age and this one is this age, roughly, if you kind of blur your eyes and you say, well, that's roughly how it is. But if you talk to Peter Matusek, who's a soil ecologist down at Stanford, and he studied this island inside out. And we went to talk to him about the age of the different substrates across the island. And, when we talked to him, we just came out tearing our hair out because he said, it depends on the ash, because if the ash falls recently, then it grounds the effect of the age of the geological age. And so, we at the end of that, I didn't know what to do. But you do have approximately all, the age structure of these volcanoes. So what we're thinking of doing now is just kind of looking a little bit farther removed and trying to see, well, this is a community that we can say is very young. This is a community that we can say is older, etc. And try and get a signature change. That was a long answer.
Well, if there are any more questions,